So then the next presentation is by Anusha Balla. Anusha is not here, so she's quite far away. She's in her home country in Nepal. And I think, yes, we have a connection there. So please, Anusha. She's coming from University of Eastern Finland and will talk about drug penetration.
Good afternoon, everyone. It is my pleasure to present my research topic, drug penetration to ocular surface tissues, today at the EuDES Symposium. I am Anusha Balla, ESR 7 from the University of Eastern Finland. Although I am not able to attend the program, I am really thankful to PAS and the IT team for providing this platform to present my research online. Now, moving on to the topic. As we all know that the topical administration is the most commonly used delivery system for the ophthalmic formulations. It is because of its ease of use and non-invasiveness. However, it is not the most effective delivery system in all the cases. And it is due to various factors such as tear turnover, reflex blinking, corneal barriers, and nasolacrimal drainage that the bioavailability of the topically applied formulations is extremely low. And here is how our project aims to improve the ocular bioavailability of the topically applied drugs. The first one is that the physiochemical properties are really important because they define the ocular surface uptake and partition of the compound. So for the first one, we evaluated those physiochemical properties. And the second one is the testing and optimizing novel drug delivery systems that help to prolong the retention of those compounds on the ocular surface. And the final one is testing the novel drug candidates for the treatment of dry eye disease. Now, moving on to the experiments. These slides show the experiments that I have done, and I will talk about the data obtained from these studies under each task, but here I will just describe these studies in brief. So starting with the evaluation of the physiochemical properties, for this we have studied the solubility at different pH, and for stability we have monitored the pH and concentration of compounds over a period of time. And we performed the studies at different storage temperatures and also presence and absence of light. Moving on to the in vitro uptake and ex vivo drug distribution studies. The cell line that we used for the in vitro uptake study was human corneal epithelial cells, and we have used porcine ocular tissues for the ex vivo drug distribution studies. In case of the in vivo studies, we have performed safety studies, which the main aim of the safety studies is to ensure that the compound is safe for topical application at that concentration and that it doesn't cause any irritation or inflammation. Next, we have also performed ocular pharmacokinetic studies. And the final one is the pharmacokinetic modeling, for which we developed some STELLA models. And these STELLA models have helped in prediction of the drug concentration in ocular surface tissues and also in the tear fluids. And with the help of these models, it will be easier for the formulation development. Now, let's begin with each task, and I will start with the first one. Although the ocular drug absorption after topical application has been studied, the spatial drug distribution of the compounds in the ocular surface tissues has not been explored much. So in the first task, what we did was we selected 7 beta blockers with similar molecular weights and different lipophilicity, as you can see in the table. and then we studied its uptake and drug distribution. We have already published the data from the study, and for more detailed information, you can go through the publication. For this study, we have performed ex vivo drug distribution in the porcine ocular tissues, cellular uptake in HCE cells, and finally, in the in vivo studies, we have performed the drug distribution study in the rabbit cornea. Now, I will start the explaining it in brief, starting with the ex vivo studies. In the ex vivo studies, we performed the study using Using chamber, and after that, after the experiment, we separated epithelium and stroma by scratching the epithelium from the cornea and did the analysis. So from the results, what we found was that the lipophilic epithelium for the lipophilic epithelium, there was a positive correlation between log D and distribution coefficient between epithelium and donor solution. The same was observed for conjunctiva. However, for the hydrophilic stroma, there was a negative correlation between log D and the distribution coefficient for stroma and epithelium. Moving on to the in vitro study, for this we found that there was significantly higher intracellular concentration of the most lipophilic compound propranolol from the beta blockers used in the study, and there was significantly higher intracellular concentration compared to the lipophilic atenolol, and we found that the results obtained from this in vitro studies was quite comparable to the ex vivo studies. In case of the in vivo studies, we used MALDI-IMS, which stands for Matrix Assisted Laser Desorption Ionization Imaging Mass Spectroscopy. With MALDI-IMS, what we did was we used 3 beta blockers from the same beta blocker, 7 beta blockers that we used before, and we determined its spatial distribution in the rabbit cornea after eye drop installation. And now, as seen in the figure, is the propranolol in the figure A. It's after 10 minutes of eye drop application. In the figure B, it's after 60 minutes, whereas C is the blank image. And in the study, we observed that the relative distribution of the compounds there was relative distribution in epithelium and stroma, and we found that the concentration seemed to be higher in the superior cornea compared to the inferior cornea. Now, moving on to the second task. The rapid precorneal loss of the topically applied eye drops limits the ocular drug absorption. So, in this case, new drug delivery system with controlled release and enhanced precorneal resistance properties may improve the ocular drug bioavailability and also the duration of action. And during my secondment at HG Beyond, the University of Santiago de Compostela, I had synthesized and characterized hydrogels, which I used to load dexamethasone as a model drug after returning to UEF. And the paper related to this studies is now under review. For this study, what we have done is the first one is the loading and release of the dexamethasone in vitro, followed by in vivo pharmacokinetic study in which we compared hydrogel suspension and solutions. We compared these two formulations, and finally we developed a Stella model for simulating the tear fluid concentration and compared it with our in vivo results. I will now start with the start with the in vitro studies. In the in vitro studies, we found that the highest release and loading of dexamethasone was found in the hydrogel with highest acrylic acid, and therefore we've selected this hydrogels for our further studies. For the in vivo studies, as I have mentioned, that we have compared these three formulations: solution, suspension, and hydrogels. And from the in vivo pharmacokinetic study, higher soluble dexamethasone exposure in the tear fluid was found in hydrogel, followed by suspension and then by solution. And we can see that the AUC from 0 to 120 minutes for hydrogels was significantly higher compared to the solution. And for suspension, we have also calculated the soluble dexamethasone by considering the soluble fraction from suspension itself. From this study, we can say that the higher precorneal retention and exposure in the tear fluid was found from hydrogel, and these factors actually indicate better efficacy of the dexamethasone hydrogel. Then for our Stella models, we have used the in vitro release rate from our in vitro studies, and we have used two conjunctival clearance values, and from these we have predicted the tear fluid concentration of dexamethasone. And here you can see that the two predicted values and our in vivo results are quite similar, and we can say that these Stella models really help us to have the prediction for the in vivo results. Now, moving on to the third task, which is testing of the novel drug compounds for the dry eye disease. We have tested two novel compounds provided to us by University of Antwerp, which are UAMC 50 and UAMC 3203. I have already presented the results from UAMC 50, for which we have performed the studies for the solubility, and also I had the opportunity to load and release to study the loading and release of this compound in hydrogels during my secondment. I will not be presenting the results for this compound, as I have already presented this in the earlier meetings. And that now I'll be focusing on the other compound, UAMC 3203. And for this compound, we have performed these studies here in Copio. The first one is the solubility and stability. We have studied the solubility at different pH, and for stability, as I have mentioned earlier, we studied the stability at 4 degree, 37 degrees, and at room temperature in the presence and absence of light, and we monitored it for a month. And we also performed and the other two tests are ongoing, which are the in vitro uptake study and the drug distribution in the ex vivo studies. And as I have told that these studies are ongoing, I'll be presenting just the preliminary results for now, and the final one is the in vivo safety studies, which I have also already presented. And starting with the solubility and stability studies, now I'll be focusing on the stability results. And as you can see that the pH was not affected much by the storage conditions and also by the presence and absence of light, and also for the concentration, the the concentration did not decrease below 90% on any storage conditions, but comparatively it is shown that for at 4 degree it was quite stable compared to the other storage conditions. And now for the in vitro uptake studies and the ex vivo drug distribution studies, these are the preliminary results. As we can see in the upper two figures, that the compound seem to stay more in the epithelium, as the concentration is much higher in the epithelium compared to the stroma. And for the in vitro uptake studies, the uptake also seems to be quite higher. But as I have said, that these are the preliminary results, and I will be conducting these again after soon. So now, moving on to the in vivo safety studies, we have performed visual inspection, OCT, and stereomicroscopy, and we found that the compound is safe for topical application at the concentration 100 micromolar, and we did not see any inflammation or irritation in the rat's eye during this study. So we can say that it's safer for topical application at this concentration. Now for the future works. I'm now, as I have mentioned, I will be working on for the permeability study of UAMC 3203. I'll be focusing on the compound, and we have also planned for joint publication with for the compound with Camila and Agnese, and we are already working on it. Regarding collaborations, I will be working with Camila and Agnese on the compound UAMC 3203 for its characterization and its effects on the dry eye disease. And also I'll be working with Bao Tran for the THS formulations, and some of the experiments have been completed here at UEF during his secondment, and also we will be working on the project. And regarding the secondment, I have secondment for University of Cologne for coming January and February, and there the experiments for the UAMC 3203 in the desiccating stress mouse model have been completed, and now we will be working on the immunohistochemistry and molecular biological studies there, and I'm really excited for the secondment there model. And finally, I would like to thank each and every one of you for the support and guidance throughout this journey, from everyone from UEF to HG Beyond and University of Antwerp. Thank you, and thank you for your attention.
Okay, thank you, Anusha. Very clear presentation. Hello. So there's a question here.
Yeah, I was just wondering, you did some of your topical dosing in rabbits to look for residents. Rabbits have a very different tear film, right? They have a Harderian gland. They secrete very dense lipids to the ocular surface, which is why they don't blink. I was wondering why you chose that model.
We use the rabbit model for the hydrogels because for also considering the size of the hydrogels, it was easier for us to use the rabbit model instead of the rats, and I think it's the main one.
So you didn't see any interference from the lipids there that confused your results at all?
I suppose not.
No? Okay.
Yeah, I guess the tear film lipids are relatively minor factor. factor in the tear film pharmacokinetics. So there are much bigger other factors, and I think the literature indicates that rabbit and human, the clearance is a little bit higher than in a rabbit, but they are pretty good correlating and not that far away from each other. Okay, any other questions? Okay, it seems that there are no other questions. Thank you, Anusha.
Thank you.